Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Quality by Design in mRNA Manufacturing, at-line monitoring coupled with DOE for higher IVT yields and efficiency of purification. I'm Lauren Coyle, an editor at BioInsights, and joining me today is Rob Sierkinik, who will discuss optimizing IVT reaction yields and costs using DOE alongside high-throughput mRNA purification to minimize dsRNA and improve the effectiveness of mRNA therapies. After the presentation, we'll have a live Q&A session, so feel free to pose your question to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. Now, I'd like to introduce our presenter. Rob Sierkinik, a chemistry graduate and PhD holder from Oxford, leads a team at Sartorius BIA Separations focused on efficient purification methods for pDNA and mRNA production. So without further ado, I'll hand over to Rob to kick off today's presentation.
Thank you, Lauren, for this nice introduction, and it's great to be back to give this webinar on everything that we've been working on since you last heard from us. Today's presentation is really going to talk about how to build in quality by design in mRNA manufacturing. And as Lauren has already announced, I will try to cover some aspects of design of experiment application in the context of IVT reaction, the reaction with which we produce, I believe, currently still all mRNA vaccines. This we will look in context of also previous developments that got us to the application of DOE, and then we'll take the IVT product that we produce in high yield through to purification. And as Lauren mentioned, my desire is to take you through several stages of purification, tailored really to the highest purity mRNA that we see today is being required for mRNA therapies, where purity is key to clinical success of mRNA. So this is what I would like to cover in the next 40 to 45 minutes. Do stay with us for some exciting updates. Before I go into experimental results, which there will be quite a few in the slides presented, I do want to pause to ask the fundamental question underlying this presentation: what is QBD? What is quality by design? And a simple or simplified answer to this would be that it's basically a systematized approach to development, meaning we do not do research for its own sake. We have a very precise set of goals where we want to get to and how, and we set out at the beginning how we plan to get there. So it's development that begins with predefined objectives and emphasizes product and process understanding and control. And this may be slightly different from how we were taught to conduct experiments in university still, because it really puts the end in mind from the very beginning. So we have, and it applies broadly, not just for mRNA, of course, it applies to all biopharmaceuticals, but even more broadly than this. It asks us, and it would be, I think, great practice to apply everywhere in life, really, is to establish project targets and goals early on before we start a project. This, I think, we mostly do in our pursuits. Define the market, the customers that we aim to deliver our solution to, discover the needs, features of the design, and so forth. So really put the end goal in mind as we begin to develop the process. And as mentioned, the biopharmaceutical industry did not come up with this design. Back in 1992, Mr. Juran, or Jaran, I don't know how to pronounce his name properly, published this seminal book, Juran on Quality by Design, for a completely different industry, but it was about planning quality into development of goods and services. And it was only later that the QBD design, QBD concept, philosophy was picked up first by the FDA, and later, of course, all of the other regulatory agencies, and started more and more to require this way of thinking in the biopharmaceutical industry. And fast forward to 2013, and there's now an expectation, and it started primarily at generic drug manufacturing to implement quality by design. Why there? Probably because there that the end goal is really known from the beginning. We know exactly what we have to make. It's perhaps more difficult when we develop new medicines, but also there we can apply similar concepts. And so I want to think a little bit about what is QBD and in medicine development or in process development, how it differs from traditional approach, and we can broadly differentiate three large segments under which we can cluster the differences in approaches. One is pharmaceutical development, which is either in the traditional development empirical and does one factor at a time approach, or OFAT. Quality by design would try to build a systematic understanding and apply a multivariate approach to understanding the pharmaceutical and process development. Second layer is process control. So we have to perform a lot of in-process controls, a lot of in-process analysis to take good go, no-go decisions. Has this particular purification step worked, yes or no? We have to measure the outcome. On the other hand, QBD will build a systematic understanding where, based on some online measurements, we can already predict where a particular experiment, particular run sits within a knowledge space, and we do not have to take lengthy measurements and make decisions because we know already where we are in our predefined space, which we'll talk about more later. And the control strategy differs between traditional and a QBD approach by testing and inspection in the olden strategy, and risk-based control strategy, meaning that because we have a better system understanding, we can better apply risk-based methodology, don't have to test as much because we know where we are based on a lot of prior knowledge. We'll be building this knowledge particularly with respect to IVT reaction in the next few minutes, so it might be easier to understand what I mean there. QBD is broken down in multiple very well-defined steps. So when a pharmaceutical development starts, with manufacturers typically start with the target product profile, which summarizes what a particular development should even target. That's based on patient needs. So what's the target molecule? What issue does it address, and how? And then that is translated into so-called quality target product profile, which forms the design of product development. So what are really the quality attributes? And then from that is defined a set of critical quality attributes. And those are quality attributes that really have to be gotten right, or else the pharmaceutical ingredient can either not be effective or it can even potentially be harmful to the patient. So we have to understand which quality attributes are critical. We'll talk about critical quality attributes in terms of mRNA drug substance in a second to make this from abstract to more concrete. Once we understand what are the critical quality attributes, we then need to control them we need to derive them through controlling our manufacturing process. And so those two are critically related. And again, we'll be building some understanding between critical quality attributes and the process for making mRNA. And then once we have that, we start defining so-called design space, which means we build ranges for process parameters, for example, temperature, pH, concentrations of certain reagents. We'll be dealing a lot with magnesium, with NTP, plasmid, T7 polymerase in context of mRNA production. And once we have the design space, we can then design the control strategy by using a risk management approach. This really means where we are, we can move between different parts of the design space surface and still remain within a safe boundaries of critical quality attributes without having to trigger certain regulatory changes and so forth. So we'll see how this applies in action. CQA, as I mentioned, is a key component of the quality by design approach. So what is a critical quality attribute? Well, it's a physical or chemical or biological or microbiological property that should be within a certain limit, a range, or distribution to ensure the desired product quality. We will be using CQA as so-called required responses in our design of experiment exercises. But before we can do that, we need to identify them. So we identify them based on so-called quality target product profile, which means we need to understand the link between particular physical chemical property of our molecule, of our active ingredient, and the severity of harm to a patient from the safety or efficacy perspective, which would, so to speak, go wrong if we fail to meet this quality attribute. We will see in the context of mRNA, double-stranded RNA is such a critical quality attribute, which needs to be minimized in order to minimize immunogenicity of our product. Yeah, and so without further ado, I do refer to I think the seminal publication in the field of mRNA, a publication by Simon Daniel and the team at Imperial College, who have published this wonderful paper on quality by design for enabling RNA platform production processes back in 2022, where they defined for the first time a set of critical quality attributes for mRNA manufacture. They defined RNA content, sequence identity, integrity, capping efficiency, poly-A tail length, and poly-A tail level as critical quality attributes for mRNA itself. So let's say product attributes. There are product-related impurities, such as RNA purity, double-stranded RNA. This is the one we're going to talk about most today, as well as RNA fragments or shorter RNA species. I will not talk about the DP attributes, meaning the LNP attributes. This is not part of today's presentation. Let's talk still a little bit about the process quality attributes. So RNA precipitates, residual enzymes, residual host cell proteins, and residual DNA were defined as those process-related quality attributes that we also have to take into account. So this is from a formal QBD approach to mRNA. Those are the CQAs that came out. Today, I will really focus on dsRNA as perhaps the most famous impurity in an mRNA process. And I will try to couple the control of double-stranded RNA with productivity of mRNA drug substance itself. So how do we make as much mRNA while keeping double-stranded RNA as low as possible? And later on, we will show that in case we do still have some residual double-stranded RNA in our product, how do we remove it to a level that is completely safe for application into an animal model or in the clinic. Before I get there, I do want to go now from this formal QBD framework mindset, if you like, to a more physical, chemical, scientific understanding of mRNA, because I think it's important for everything that we're going to be discussing later on. So what is mRNA to a chemist, to a scientist, before we start formalizing the production in terms of QBD? Well, mRNA is actually quite unlike any other biopharmaceutical used in clinical use at the moment. Its major feature, I think, if I highlight one, is it's huge. It is at least 10 times higher than the next largest biopharmaceutical in widespread use, which would be monoclonal antibody. Monoclonal antibodies are about 150 kilodaltons. The smallest mRNA produced in labs, which would be a humble EGFP, is already over a megadalton large. COVID vaccines are 4 times larger than that. So we're dealing here with a really extremely large biomolecule that we have to produce at scale. Second feature, probably also often overlooked, is the fact that it's a very acidic molecule. Its pKa is very low, and that makes it challenging in purification processes. We have to completely adapt purification processes that are using more purification approaches tailored towards near neutral molecules to something that's very strongly acidic. mRNA is also prone to forming hydrophobic interactions, particularly when in contact with salt, which, when gone badly, means it can form precipitation. But when used wisely and to our advantage, it can actually be used as a selective purification approach. Finally, it's a wonderful feature of mRNA that it has to be polyadenylated at the point of administration for the reasons of in vivo stability. But to a chemist or to a biochemist or to anybody producing mRNA, it's a great feature that we can use for selective purification. We have a ligand called Oligo DT that can selectively capture the polyadenylated and take mRNA out of a mixture of other impurities. The last and most important feature, again often overlooked, of mRNA is the fact that it is produced essentially by a chemical reaction. And when you come to think about chemical reaction, we need to think about building blocks. And so that's why I have this lovely house built out of bricks, plastic bricks. I have small children, they love playing with those. And they know that if they are to build this house, they actually need to provide enough bricks for their play. And so this is actually a fundamental underlying feature of the work that I'll talk about today, is we need to think about making mRNA in terms of putting the bricks together to build a chain, in this case. And it's a feature that I'm often surprised how little this fact is still appreciated in the field. But we're here to change that. So mRNA is produced by a chemical reaction. It's an enzyme catalyzed chemical reaction, polycondensation of nucleotides or NTPs, guided by a DNA template, typically a linearized DNA template, catalyzed by an enzyme called RNA polymerase, typically T7, to form a chain of nucleotides, and in turn producing something called pyrophosphate. For each NTP that goes into the chain, we produce a pyrophosphate molecule. We also need to provide magnesium into this reaction. Sometimes, quite frequently in fact, we provide something called capping reagent. We don't absolutely have to provide, but it's beneficial to the reaction if we provide pyrophosphatase, because pyrophosphate, which is this side product of the reaction, is actually very poorly soluble, so it can lead to a lot of precipitation. And still, I think oftentimes we see reactions that are reported as precipitated, and it's probably because of pyrophosphate that's being formed during the reaction, not actually due to mRNA itself. mRNA turns out to be actually quite soluble, even to a very high concentration. We can add RNase inhibitor, spermidine, DTT, and other buffer components. So it's a reaction that comprises multiple reagents. We can count, depending on the design, 13, 15 reagents. So it's a complex reaction that we need to then study carefully, because as you can well imagine, a lot of these reagents have synergistic effects on the output of the reaction. IVT reaction also produces an unwanted side product, and that is double-stranded RNA. So more or less same principles as guide the production of mRNA also guide the production of double-stranded RNA. What are the exact factors that drive the production of dsRNA is still under intense exploration. We understand quite a lot of the features, but I think not yet all. And it's quite a complex field, still unexplored fully. and the limitation, I believe, and this is a call for help really, is also understanding of the quality of double-stranded RNA, because it seems to come in at least 3 flavors: 3' extended, abortive transcript double-stranded RNA, and as well as full antisense double-stranded RNA. So it's not just that we have one type of this impurity, there's multiple different categories of this impurity, and there are multiple factors driving it. We will address a few factors that we can control in an IVT reaction, but there are multiple others that we cannot control in IVT reaction, for example, sequence design and so forth. So how do we monitor this reaction? Well, we use traditionally immunoblotting to control double-stranded RNA. This is a J2 dot blot shown here at the bottom left. Typically, we've used electrophoresis, so electropherograms, agarose gels, and so forth, to measure the size and the integrity of RNA. But more and more, we use chromatographic analytics to give us better and faster insight into what goes on first in the IVT reaction and then in all of the downstream steps. Our favorite method is a method based on a so-called CIMAC Prima S analytical column. It's a monolith-based analytical column that we produce and that we have developed here in Sartorius via separation, which has a very unique feature for reasons that I will not go into, but that are typical for monolith chromatography, in that it has very high resolving power, and it has high resolving power for large biomolecules. It means that we can capture mRNA and elute it in a very short space of time. And because of the high resolution, we can separate out the nucleotides that are also in the reaction. We can, in fact, also separate the capping reagent as well as the DNA. So we can more or less see the whole reaction on a plate. And what's best about it is that we can do that in somewhere between 3 and 7 minutes, depending on the method that we take. We've developed multiple versions of the method, differing a little bit in length and resolution, but between 3 and 7 minutes, we can get a resolution of all nucleotides, DNA, and mRNA. And this, as you can imagine, is very powerful when dealing with IVT reaction. How we use this? Well, we have this method built into analytical system, analytical platform called PathFix. It's a wonderful and very powerful analytical tool that we design in this particular segment, particularly at mRNA analytics. So really to help the field advance the understanding of mRNA processes, we make it very easy for scientists as well as operators with pre-programmed analytical protocols to really make it as easy as possible to obtain critical information about their analyte. And what does this information look like? IVT reaction, as we said, combines NTPs into mRNA chain. And if we have good resolution between mRNA and NTPs, then we can monitor the reaction in near real time. If it takes 3 minutes to read off these concentrations, then with a 3-minute lag, we can see what's going on in the reaction. So this is a typical IVT reaction. mRNA is being produced whilst NTPs get consumed. This is a graph. It's an interpretation of chromatograms, and here we see what our analytical system would actually see. We see mRNA peak increasing whilst NTPs decrease. Capping reagent, this can be ARCA, it can be any other capping reagent, we see staying more or less constant throughout the reaction. It doesn't get used up. Now you can imagine what you could build with this information. First thing, and this is what we published on quite a few years ago now, well, in fact, 2 years ago, a lot has happened since, is we can monitor the effects of individual components of the reaction on the rate of mRNA production. So, for example, magnesium. What's the effect of magnesium on the rate of T7 enzyme or on the rate of mRNA production? What's the effect of plasmid DNA or NTP concentration on the T7 rate, and so forth? You can now start asking many, many questions, and you start getting back time-resolved information about how mRNA is being produced and how NTPs are getting consumed. This is one example. This is mRNA being produced. Here we paused production because we took magnesium out, and then we put magnesium back in. We wanted to show what happens with NTP consumption. Here, this is the same story where NTP consumption is being shown for the same sample. We see that as soon as we took magnesium out, NTP consumption also stopped. Once we put magnesium back in, reaction resumed, both in terms of mRNA production and NTP consumption. So this is a very powerful tool to start asking the reaction, what affects you, or how would you feel today, so to speak. So one of the ways in which we can utilize this information is by convincing machines, bioreactors, to start performing IVT reactions for us. In this particular case, we programmed AMBER 250 bioreactor to start dosing NTPs into IVT reaction in an automatized fashion. If you're interested in this, this is published in Chemie Engineer Technique also about 2 years ago now, of how we utilize at-line monitoring to basically significantly increase the productivity of IVT reaction in fed batch mode. This is what the results look like. This is not maybe the main topic of today's webinar, but an important side use of this methodology. What I really want to tell you about today is the story of how we can now apply this immensely powerful analytical tool, which is able to separate mRNA from nucleotides, from DNA, from capping reagent, to build a knowledge base of the IVT reaction by coupling analytical technology with design of experiment software approach. And so we coupled PathFix analytical tool with MODE software to build, I dare say, one of the currently only comprehensive DOE sets published. This work is all published in Biotechnology, Bioengineering, so if you want to read the full story, but I will take you through some of the key findings of this study, which I should say is an example of how a DOE study can be conducted. It's not to say that this particular result set applies to every single mRNA that you produce. So my suggestion is to listen to the following slides in sense of inspiration of how you can approach your development of mRNA program. You can see already from this slide that these are excerpts from experiments conducted in MODE, how different IVT profiles we can obtain. This is always mRNA being produced and NTPs being depleted, and you can see already how much we can affect mRNA production by changing process factors. So first, when we set off the development, we wanted to set ourselves a threefold aim. One is maximize the yields. Second, minimize the impurity, which is the double-stranded RNA, and maximize cost efficiency. And so we wanted to build a knowledge space within which we find a design space within which we can move in terms of the process, and also then find an optimal set point for our process. The first part of the exercise was actually to do some reading. There are multiple prior publications which looked at the effect of process factors on mRNA output. We found that the space which gave the best results, particularly in terms of NTP and magnesium to NTP ratio, was actually relatively narrow, because most of these experiments profiled here in quite a breadth of conditions didn't actually yield very much RNA or didn't yield very pure mRNA. So we decided to define our knowledge space as this. And within that knowledge space, we would look for our design space and find the optimal set point. This was the design of the study. I'm not going to go into details, but they're based basically on prior literature search. So reading prior literature is always a good starting point. And then we converted the design, so a theoretical design, into performing IVT reaction. And at each point, when we were measuring IVT reaction for up to 3.5 hours, and every 15 minutes we took a measurement, and we could read off for every time point what's the mRNA concentration, what's nucleotide concentration. We also measured dot plots for every time point, and so we could build time-resolved models and iterate, and this way build a design space. One thing that we also defined very early on, which I think was critical to the success of the exercise, was first the understanding that mRNA production is a chemical reaction, and as such is governed fundamentally by stoichiometry of the reaction, meaning we need to understand how many moles of reagents can turn into how many moles of product. We need to think as chemists. A young man called Jimmy Bowman, who was the lead author on this work, then came up with this approximation, which seems to apply quite generally, and I want you to be aware of it because it's very powerful, that 1 millimole of limiting NTP gives rise to about 1 gram per liter of mRNA. 10 millimoles of NTPs give rise to 10 gram per liter, and so forth. And so we like this equation so much, we call it the Bowman equation after Jimmy, who came up with it. And so then we start building our models. Because we treated the reaction as a stoichiometrically driven process, we could quickly find that our first model was already giving us some good indications of what influences the reaction, what doesn't. But we could see that the model started to give us indications that the yield was being unrealistically high, over 160%. If we did not take the stoichiometry into account, we would probably miss this. And so we said we needed to bring in more model terms in second iteration, which helped us decrease this maximal predicted yields to 120%, still more than it's actually chemically possible. And so in the third iteration, when we then fully modeled the reaction, we got the yield maximal predicted yields to 105%, which was already within the error of the method. And so we were happy then with this particular model. You could see that we modeled T7, DNA, magnesium to NTP ratio rather than magnesium and NTP individually, and we modeled NTPs. You can find out the whole model in the publication. So what we learned was that in order to target the desired mRNA yield, we needed to set the target concentration of NTPs. That's the 1 millimole takes us to 1 gram per liter. 10 millimoles took us to 10 gram per liter. We saw that higher NTPs resulted in lower reaction rate, and we also found, this is quite important, that optimal magnesium to NTP ratio decreases as NTP concentration increases. And so we found that a ratio of approximately 1 is good, but as we increased NTP concentration to even higher levels, we had to decrease this ratio a little bit. We also looked at double-stranded RNA, and to cut the long story short, what we found was that our model was agreeing in a certain respect with previous publications that magnesium affects double-stranded RNA production, but we took it a step further to show that low magnesium to NTP ratio actually results in low double-stranded RNA. So as long as we kept the ratio low, we could actually take NTPs to a high level and thereby get high yield whilst getting low double-stranded RNA. And very few other factors actually had significant effects. We then validated this model. So we didn't just leave it there in the theoretical level, but we actually validated that the model works. We verified the predicted optimum, and we showed that cost efficiency of our final optimum was actually about 40% less than the cost of the reaction before we started the optimization. And so this was our optimal set point, which we derived for our reaction design. We validated the model on longer constructs, 4,000 nucleotide construct, where we were predicting also the kinetics of the reaction. And you can see that the measurements agreed very well with the prediction. So the model seemed to apply broadly. Interestingly, the double-stranded RNA model did not apply for the longer construct, and we believe that that's because the constructs were actually from different construct families, from different plasmid backbones with different UTR designs and so forth. So we may need to do further work to understand the generality of the findings. And finally, and this is the piece of work that we're very proud of, because we said if we really now claim that we understand what drives this reaction, we should be able to take the yield of IVT to where it's never been before. So we said if there's a stoichiometric relationship between NTPs and the yield, then let's try to double the previously reported highest yield, which was about 12 gram per liter. Let's target 25. And so what we needed to do was put in at least 25 millimoles of NTPs. We had to adjust a few other things that are listed here and also described in the paper. And gratifyingly, the reaction went all the way up to 25 gram per liter, taking us into unprecedented high yield environment, also with completely different cost efficiency, which needs to be now further explored because it's quite attractive. We also validated that we could recover all of this RNA in the purification, and so we put it through Oligo DT and confirmed that we could recover an equivalent of 25 gram per liter of mRNA. So we double verified our analytical determination of the content, and even more gratifyingly, the reaction was performed in a way that we predicted would also lead to low double-stranded RNA, which indeed it did. So this was a very successful experiment, which I see would become very interesting when the cost pressure on mRNA production becomes even more aggressive than it is today. And so I believe that we will return to this approach sometime in the future. Now, I did promise that I will speak also about purification. So I touched upon this already in trying to recover this 25 gram per liter from the IVT. How we did this was through Oligo DT. Oligo DT is the driving force of the mRNA downstream processing. It's an affinity ligand that captures the poly-A tail, and so it's very selective. We need to put the mRNA in contact with some sodium chloride, about half molar, and then elute it with low conductivity buffer or even just water to recover highly pure mRNA with high recovery. Now we can apply DOE approach to purification as well. We published a small piece in 2022 in Bioprocess International on how we utilize a 96 well plate Oligo DT monolith format shown up here, operated with vacuum pump or even just a centrifuge to explore what factors govern the yield and the binding capacity. And so we could show the effect of salt, for example, on the binding capacity and on recovery. I will not go into too much detail here. Safe to say that the same methodological DOE approach can be applied to screen multiple factors, and a 96 well format is very powerful here because it allows us to do multiple experiments at a time. Interesting finding at the time was that we could replace sodium chloride with guanidine and get even higher binding capacities on Oligo DT, and we could hit 6 gram per liter binding capacity by using guanidine hydrochloride. We could translate then from 96 well format into column format, keeping and showing that the reproducibility was there in terms of binding capacity and recovery. And so we sort of validated that 96 well format is a good scale down model also for preparative monolith Oligo DT columns, which can take us then all the way up to 40 liter bed volume, meaning that we can produce really multi-gram amounts of mRNA in a single chromatographic run. We always couple the two approaches, the analytical and the preparative, hand in hand. So there's always in our labs a purification column sitting next to an analytical path-fix system, and we use now the combination of those two, shown here in the top right. by this point you already can read with me the fact that these are nucleotides and a capping reagent. This is our RNA, this is the plasmid. And so with the same analytical platform, same analytical method that we previously used to optimize IVT yield, we now use to monitor, for example, nucleotide and DNA clearance from our IVT reaction. And so we show here that we load the column. In this case, it was actually a multimodal column. It's a story for another day. Loaded RNA that also had NTPs still present. All the NTPs went straight through the column, did not bind. The plasmid bound, but was washed off, and the elution contained pure saRNA in this case, actually. And so this is how we couple the at-line monitoring philosophy, which is for us at the heart of quality by design for mRNA therapy development. And let me finish on the thought that IVT also produced double-stranded RNA as the unwanted side product. So how do we deal with this? Oligo DT turns out to really remove quite a lot of double-stranded RNA impurities, presumably those that are not polyadenylated, but typically there would still be some residual double-stranded RNA at that point. And so the way we deal with that very reliably is by the use of reverse phase ion pair reverse phase chromatography. First postulated by Katalin Karikó all the way back in 2013 as being very effective, and indeed it's super effective. It's also very scalable, and we've been developing approaches for both mRNA and self-amplifying RNA in depleting the RNA of the double-stranded RNA impurity. We can operate reverse phase column. In this case, it's called SDVB, styrene divinyl benzene, in an acetonitrile gradient, either in linear gradient, where we always find the central part of the molecule is double-stranded RNA free. Once we have a good understanding of this, we can turn this linear gradient into a step elution, making it even easier to process. We just elute short double-stranded RNA fragments here. Main elutions always double-stranded RNA free, and then the final strip fraction contains the long double-stranded RNA molecules. But it gets better than this. We've shown that we're not only removing double-stranded RNA with this step, we also remove fragments as well as residual DNA template. We developed a whole analytical method around monitoring residual DNA clearance, which I'm not going to go into. It's published here in Analytical Bioanalytical Chemistry. It's a PATFIX method that monitors the linear DNA impurity in our RNA product. And what we were able to show very convincingly is that reverse phase chromatography is as good as DNase in removing residual DNA template. And we do this across scales. We've shown it for small scale. I show here a purification of 100 mg of mRNA on an 80 milliliter monolith, which looks just the same as on a 1 milliliter unit. We've cleared double-stranded RNA in step. We've also cleared residual DNA template without having to use DNase. So we saved the cost of adding DNase, an extra enzyme. We didn't have to show DNase clearance there. And so in the same step that also removed double-stranded RNA, we removed residual DNA template as well. So very powerful tool that works very reliably. And to show that it works also in cell-based context, we work together with Tron, and I'm very grateful for these data sets on self-amplifying RNA that showed really a staggering increase in cellular activity, in this case of self-amplifying RNA, so the most challenging of systems of RNA products. You can see here quite a staggering difference between before and after reverse phase purification in terms of activity of this RNA, as well as in the interferon response. So the response of RNA that was treated with this reverse phase chromatography was very, very low compared to non-SDVB treated self-amplifying RNA. And then This was after 6 hours, after 24 hours, the innate immunogenicity of this RNA became apparent, and so the one-step versus two-step purification didn't make as much difference. Anyway, you can look forward to reading this in a paper shortly. For now, I have to thank you for your attention, remind you what you've seen. So I really wanted to convey the importance of quality by design approach in terms of mRNA, and hopefully I've shown an interesting case study of how we use DOE as a core part of QBD approach to really get a good understanding of the IVT reaction from the process perspective, how we were able to reach unprecedented high yield of IVT, 25 gram per liter, purified the mRNA with affinity chromatography, and then polish it off with reverse phase to really have a high yielding, but also high purity, I'd say extraordinarily high purity of mRNA as well as saRNA product. Very happy to take any questions. I thank you again for your attention. I'm always glad to take any questions either now or then by email. And I also am obliged to a wonderful team that works on all these very interesting challenges in mRNA field. Thank you again.
Well, thank you, Rock, for a great presentation there. And we are now going to start our Q&A. So I'd like to invite you on camera with me and we can get started. Fantastic. So our first question we have here today is, can any RNA modalities be followed in IVT?
Yes. So we have explored quite a wide variety of IVT-derived RNAs. Most recently, tRNA, which is much smaller than mRNA, so only about 75 nucleotides. We've in fact used the same analytical approach to monitor IVT, which has been published in Frontiers Molecular Biosciences. So that's sort of the smallest IVT product. mRNA I spoke at length about, and then the largest IVT-derived RNA would be self-amplifying RNA until somebody comes up with a larger one. So with the same identical analytical approach, we cover RNAs from 75 nucleotides all the way up to 10,000. So it's quite a powerful tool, and I now look forward to tRNA therapeutics, which are slowly coming up as well. Also start to consider the same approach as mRNA industry is now taking in terms of this IVT monitoring and optimization through the tools that I described today.
Wonderful. Thank you so much. And our next question here is, does viscosity of the IVT cause any difficulties for purification, such as high pressure?
So one interesting finding was that IVT viscosity really does increase significantly when there's more mRNA produced. So at 25 gram per liter, we are dealing with a very highly viscous solution, which from the process perspective may pose certain challenges. However, we were able to purify all of the RNA through an affinity Oligo DT monolith without any issues. There was a dilution step in between, so the IVT, as it always is, even in lower yield systems, even if your IVT yields 8 or 10 gram per liter, you typically always dilute the reaction before loading onto Oligo DT. And so we did much the same, even for a 25 gram per liter reaction, and Oligo DT run performed completely as expected. So it's not to say that certain RNAs are not more prone to aggregation precipitation. This should be addressed during process development. We do see that certain sequence designs are more prone towards forming precipitates, and this does need to be addressed. In fact, Oligo DT 96 well plates we find are very useful for monitoring the propensity of RNA towards precipitation, because it's a vacuum operated system. If there's any precipitation in the sample, the plate will detect this. It'll make it more difficult for the sample to pass through. But we find that this is more somehow linked to the sequence design and to actual process. So you can have two RNAs. One will be prone to precipitation and therefore to causing more process trouble. Under identical conditions is another one which will behave beautifully. So this is a word of caution when designing sequences, to have this processability of the RNA sequence in mind, and to address it early. And yeah, I can commend these 96-well plate Oligo DT monolithic plates as actually a fantastic tool to compare propensity of RNA to precipitation.
Wonderful, that was great. Thank you so much. And then wrong question there. What is a typical binding capacity for Oligo DT?
Yeah, so we've written and explored this at length. We've pushed it all the way up to 6 grams per liter. We would typically see the range of 4 grams per liter, something like this, depending also on the RNA length. The longer the RNA, the lower the binding capacity. This is well understood by this point in the industry. And then salt loading concentration plays a very big impact on the binding capacity that we can achieve. So the more salt you add at the point of loading, the higher the binding capacity. But there's an optimum there. That's why the design of experiment approach is really powerful here, because there will be too much of a good thing, at which point the RNA starts precipitating. But if you're in that sweet spot where RNA is still not prone to precipitation, you will be able to hit relatively high binding capacities.
Fantastic, thank you. And can Oligo DT be used for saRNA?
Yes, it can. Self-amplifying RNA is typically also polyadenylated, just like mRNA. It has to be borne in mind the poly-A tail length, which is typically engineered with the same length as for an mRNA, now sits in a much bigger molecule. So the Oligo DT ligand has to find a relatively smaller part of the whole molecule. And you can imagine we're talking now in over 10 megadalton molecules, of which we have to find 100 nucleotide poly-A tail. But Oligo DT does find it. So we purify a lot of self-amplifying RNA with Oligo DT, and it's a very straightforward tool. Yeah, it's a well understood tool, very powerful. So I highly recommend to use Oligo DT for self-amplifying RNA. Having said that, we're also developing multimodal ligands. Quite unique. One product is called Swiper, which applies multimodal approach to self-amplifying RNA purification. And that's applicable even in case where this molecule will not be polyadenylated. So that it's not an approach that's targeting the poly-A tail. It targets more the charge on the molecule, which, as mentioned a few minutes ago, RNA is extraordinarily negatively charged. So we were pioneering, I think, ligand design for high recovery of RNA. And I'm very proud to say that even for self-amplifying RNA, we can get 85% or so recoveries from a multimodal column, which I think is quite fantastic.
That was perfect, thank you. And are there any differences when purifying self-amplifying RNA compared to mRNA?
Self-amplifying RNA tends to be more fragile, relatively speaking. It's a much longer molecule. There is this stochastic hydrolysis inherent to RNA chains, and so the longer the RNA, the more chances there are for autohydrolysis, even in absence of chemical reagents that would cause hydrolysis, like temperature, magnesium, for example. Those are agents that are known to cause hydrolysis. So the fundamental principle between mRNA and self-amplifying RNA is the same, but where we see differences is that we have to be relatively faster when purifying or when performing downstream, and probably also the IVT on self-amplifying RNA. We want to expose the self-amplifying RNA to high temperature and to metal ions for as low amount of time as possible. With mRNA, we have a little bit more leeway. And so, there are differences, although they are from the. If you were just to look at the process description, it could look the same, but the time there, you would really want to shorten the processing time for self-amplifying RNA, or else risk unwanted fragmentation.
Wonderful, thank you. And I think we have time for one more question. So does the CI-MAC Prima S distinguish between modified and unmodified nucleotides?
Great question. We have examples now of chromatograms where we can separate modified UTP, for example, from wild-type UTP. Our analytical department here has been doing a fantastic job. So there's various combinations of nucleotides that we can already separate out. I would defer to my colleagues from analytical team for more details, but the first pass answer is yes, we can separate at least some, and those I think are the most relevant modified nucleotides from the wild type sort. So this gives us even more analytical power when it comes to looking at IVT reaction.
Wonderful. Well, thank you, Rock, for answering those questions, but that is all we have time for today. So any questions we didn't get to, we will reply to by email. This webinar will be available on demand tomorrow, so look out for an email from us with the link. And we'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources tab on the right-hand side of the screen or in the on-demand email. So all that's left is to thank Rock once more for a great presentation and a wonderful Q&A. And thank you to you, our audience, for listening. We hope you'll join us again soon.